The incorporation of CoolCell modules and ThawSTAR system in a GMP facility for the manufacturing of regulatory T cells for the use of autoimmunity and transplantation rejection. I'm Amy Putnam, and I work in collaboration with UCSF Diabetes Center and Transplant Surgery, leading a Treg manufacturing team that supports clinical trials in autoimmunity and transplantation. As an overview to our justification for moving these cells to the clinic, I would like to focus your attention on two figures. The first one in the upper left shows the importance of Tregs in an autoimmune setting using an NOD mouse for a model of type 1 diabetes. We show that a normal NOD mouse goes on to develop diabetes at about 15 weeks of age, and that an NOD mice depleted of Tregs gets diabetes at an accelerated rate, shown in white. NOD mice treated with Tregs prior to disease onset do not go on to develop disease, as indicated by the green symbols. In the next figure, we show that once an animal develops disease and left untreated, it will not go into remission, shown in red. However, treatment at the onset of disease results in quick remission in about 60% of the animals, shown in green. This led us to the belief that we can do this in clinical trials, and we recently finished our type 1 diabetes phase 1 clinical trial treatment in October 2013. This data is in the process of being submitted for publication. At this point, we also have a type 1 diabetes follow-up study, a lupus study, and a kidney transplant study. Justification for alloantigen reactive Tregs. In the preliminary studies, we showed that donor B cells grown using a combination of IL-4 and CD40 ligand, SBCs were generated. These SBCs were put into culture with CFSC-labeled recipient PBMCs and performed an MLR assay. The scientists performing these initial studies noticed an increase in CD8 proliferation, as well as an increase in CD4 proliferation, but what was most striking was the subset of CD4 cells known as Tregs. This led us to the justification that we're ready to do this in patients using stimulated B cells with IL-4 and CD40 ligand to generate potent alloantigen reactive Tregs for potential clinical use. At this point, we have 2 kidney and 2 liver trials underway. The polyclonal manufacturing process is shown here, where CD4 127 negative 25 positive Tregs are isolated using cGMP antibodies in a flow cytometer and gated on CD4 cells shown here. We currently start with a unit of fresh blood, and ex vivo expanded Tregs are infused fresh. Moving forward, however, it would be advantageous to be able to freeze and thaw on both ends of the process, for example, starting materials for the Tregs, as well as following release assays prior to infusion. At this point, any Tregs used for mechanistic assays are frozen using a CoolCell and ThawSTAR CS5 provided from BioLife Solutions for non-release mechanistic assays and downstream functional tests. The alloantigen reactive Tregs are isolated in a similar way as polyclonal expansion. However, the initial stimulation is with stimulated B cells generated from either spleen or whole blood and grown in the presence of IL-4 and irradiated feeder cells expressing CD40 ligand and combined with sorted Tregs. These cells then undergo a secondary polyclonal stimulation using either anti-CD3 or anti-CD28 beads to generate clinically relevant numbers. In the future, it would be advantageous to be able to freeze and thaw these cells at various time points, including the starting material for SBCs, starting material for Tregs, previously cryopreserved feeder cells, as well as ex vivo expanded SBCs. In the future, it would obviously be very advantageous to be able to freeze cells prior to infusion after release assays. Again, these cells are frozen for mechanistic assays using the CoolCell and ThawSTAR CS5. In this slide, I've highlighted areas where the CoolCell can be integrated into the process, and I will show one example using starting material for Treg manufacturing. This particular slide illustrates that multiple leukapheresis-like products were processed into PBMCs and frozen using either the CoolCell or a controlled rate freezer. In both cases, cells were frozen at approximately 100 million cells per ml in ThawSTAR CS10 from BioLife Solutions. Vials were stored in liquid nitrogen for approximately 2 to 3 weeks and thawed the same day using a water bath. The CoolCell and the controlled rate freezer are comparable in both viability and recovery. In this slide, I've highlighted areas where the ThawSTAR can be integrated into our process, and I'm going to show 2 examples with starting material from the SBC manufacturing, as well as ex vivo expanded stimulated B cells. In this case, multiple vials of splenocytes were frozen from a single spleen. Splenocytes were frozen at approximately 20 million cells per mil in CS10 and stored in liquid nitrogen for approximately 1 month. Triplicate vials were thawed on the same day using either a water bath or a ThawSTAR. Viability and recovery is similar in both cases using both sets of instrumentation. However, it is important to note that the CFT transporter was not used and not available during this particular validation. In this slide, the spleen was obtained from 3 different individuals, processed and frozen on different days, and stored in liquid nitrogen. Depending on the date frozen, splenocytes were thawed 2 to 4 weeks later and expanded using IL-4 and feeder cells as previously shown. Ex vivo expanded SBCs were frozen at approximately 5 million per mil in CryoStor CS10 and stored in liquid nitrogen for approximately 2 weeks. These cells were then thawed either using the ThawSTAR CFT transporter system or a water bath alone. Although there is donor-to-donor variability shown in this particular slide, the trend is similar when comparing the ThawSTAR to the water bath in each individual donor. And the combination of the ThawSTAR and the transporter performed better than using a water bath alone, when following manufacturer's recommendation for the acclimation of the transporter prior to thawing in the ThawSTAR. So in summary, the CoolCell performs similarly to a controlled rate freezer with regard to viability and recovery when freezing leukapheresis-like starting material at high concentrations in CryoStor CS10. The CoolCell is less expensive than the controlled rate freezer, does not contain liquid, can be acclimated quickly for fast turnaround time, and is transportable. The ThawSTAR is comparable to water baths for thawing splenocytes. ThawSTAR and CFT transporter system performs better than using a water bath alone in a series of individual donor tests. ThawSTAR system is low maintenance, water-free, transportable, easy to clean, and therefore can be incorporated into a GMP facility for clinical manufacturing. This set of experiments shows that both the CoolCell and the ThawSTAR system can be implemented in a GMP facility for clinical manufacturing, and I've acknowledged the individuals completing this. I'd also like to acknowledge BioLife Solutions for providing the CryoStor CS5 and CS10 cryoprotectant, as well as BioCision for providing ThawSTAR and CFT transporter units, as well as the CoolCells for our testing. This work has been conducted in collaboration with the UCSF Diabetes Center and UCSF Transplant Surgery. I've shown 2 references, one for polyclonal Tregs and the other one for alloantigen regulatory T cells and our validation processes. Thank you.